Good afternoon, everyone. My name is Destiny, and recently this fall 2022 semester, I participated in DNA barcoding research with Professor Odapalli. Our research question is, what are the relatives and ancestors of the Florida plant species that I collected? Our hypothesis for this research is there may not be close relatives for our null and alternative hypothesis. I hope to find close relatives with medicinal benefits. For this research, I did research in the lab, doing bench work and on Zoom. So firstly, to start our lab bench work, we collected a plant sample from a new lake on Valencia West campus to start our bench work. And starting into the lab wet work, we cleaned our sample with 10% bleach solution and crushed it with a pestle in an Eppendorf tube. And as you can see at the bottom of the picture, this is the tube with the leaf sample tissue and the pestle that we used to crush the samples. So where is the DNA that we want? This, as you can tell, this is a plant sample or plant cell, and the plant cell is covered by a cell wall. The cell is 100 micrometers long, which is 1 million times smaller than a regular meter. And the DNA that we want is held in this ovular shaped green cell called a chloroplast. The chloroplast is packaged by two plasma cell membranes, and in order to reach the DNA, we have to then break down those layers of packages and by using lysis solution, vortexing, and a microcentrifuge. Here is some of the equipment that we use for our DNA extraction. We used a micropipette, which is a precise measurement to dispense and remove small liquids from the Eppendorf tube, and a microcentrifuge, which you place the Eppendorf tubes into, and it spins at high velocities to separate your DNA from your proteins and lipids, making it easier. Next, we mixed our DNA with a material called silica resin. And after mixing it with the silica resin, we centrifuged it to isolate the DNA. As you can see in the photo, the liquid in the Eppendorf tube is a beige and tan color. That is due to the fact that DNA is actually colorless, and when you mix it with the silica resin, it causes a beige tan color. Next, we started a Polymers chain reaction for our sample. And as you know, Polymers chain reaction creates copies of the sample. This is green due to the fact that we use a PCR master mix, which holds a green dye on the inside. After the processes of our PCR, we sent our sample to GeneWiz company, and they gave us our gene sequence. We placed this gene sequence into a website called DNA Subway, and after, DNA Subway produces a phylogenetic tree that shows the ancestors and relatives of our plant sample. This is the given plant sample, and as you can tell, there are letters N, G, C, T, and A. A, C, T, and G are all nucleotides, which are the building blocks of DNA. But N's are nucleotides that could have not been identified by Gene Subway and therefore have to be trimmed when using DNA Subway. This is the sample barcode, and we use the alignment tool on DNA Subway. This here, 25M13, is actually the sample that I have, and this is its barcode. From our barcode, you can see that it can be closely related to celery, fern, sunflower, and different species of roses. For our last part, we created the phylogenetic tree using DNA Subway. And from our picture, our plant is mostly related to polyacrofoliaceae, and a tomato plant. For the analysis and discussion, we will be taking apart that during the next semester, and we will continue doing more research. My conclusion is that I accept my alternative hypothesis that states I hope to find close relatives with medicinal benefits, and I reject my null hypothesis that states there may not be any close relatives. I would like to thank Professor Odapalli for allowing me to participate in this research and guiding me through the whole entire thing. I would like to thank Dr. Brenda Schemper for creating this research and continuing it and allowing us to use her grants. I would like to thank Valencia College for letting us use the lab equipment, and also DNA barcoding manual that held all of the protocols and steps for DNA barcoding and PCR. Here are my references. And thank you so much for attending my presentation. I invite and welcome you to ask any questions, comments, or concerns that you had about my presentation. And thank you for your interest in research.